Hi, my name is Katharina Volz. I'm the founder and executive chairman of Occam's Razor, and it's exciting to meet you today, Doris. I would love for you to just tell a little bit about your background and the research that you're doing. And
yeah. Sure. I'm a professor at UC Berkeley of Neuroscience, and I'm also an investigator in the Howard Hughes Medical Institute. And my central interest is understanding brain mechanisms for representing the visual world. In other words, how do neurons create our consciousness of the visual world?
That's fascinating. So all of this work that you've done on face processing, what has that taught us?
Yeah, so faces are this incredibly important, complex stimulus. And I think one of the things we've learned is, what code does the brain use to represent a high-level visual stimulus? And it turns out that the insights we've learned from faces actually generalize to all objects. So we've learned about the coding principles. We also know that these faces are represented in specific pieces of the brain called face patches. And so we've also, from that, we've learned about how other objects are represented, how they're laid out on the cortex as well.
Amazing. And so these face patches, are these like little islands of cells, or what are they?
Yeah, exactly. They're like small islands, a few millimeters wide, and there's 6 of them in each hemisphere of the macaque brain. And each of them consists almost entirely of these face-selective neurons that just go off when the monkey sees a face.
Wow, that's fascinating. So what kind of technologies do you use to actually see those islands, and how do you study?
Yeah, so I think this work is a really good example of the interplay between human and animal neuroscience. Mm-hmm. So face areas were actually first discovered in humans using functional magnetic resonance imaging, which measures blood flow, right? So if you show a human pictures of faces and other objects, you find these face areas light up selectively to faces. And then when we do the same thing in monkeys, so we also use fMRI in the monkeys, we see these face patches light up. But then in the monkey, we can go in and record with an electrode, which lets us pick up the electrical activity from single neurons and really study the detailed codes that the brain is using to represent faces. Yeah.
Wow, that's really incredible. What got you excited to get into that kind of research?
Yeah, I mean, I've always been interested in vision. When I was in high school, just the realization that the world is not exactly what it seems, and it's actually created by our brains and so on. So that's how I got interested in vision. But specifically, this problem of face perception was really an accident, a lucky accident. I showed these monkeys pictures of faces and other objects just like as a lark, right, to see if we would see the similar areas as humans, and then it just took me on this 20-year detour.
Yeah, wow. Actually, is this a good question to ask something like, because I know a lot of people have that problem of forgetting faces.
Yeah.
And I don't know, is there a question I could ask around that?
Yeah,
would
you
be
could
relevant?
ask, or you could say, are these face patches involved in memory of faces? I don't know.
Okay.
Since a lot of people.
Yeah. Okay. So are these face patches involved in memory of the faces?
Yeah, we actually just published a paper about this, and super interesting. It turns out that the face cells use 2 different codes for representing familiar faces. So initially, the cells use the same code for representing the features of all faces. It doesn't matter if they're familiar or not. And then slightly later in time, the code changes for the familiar faces, so they're optimized for representing the memory of faces. So yes, it's a beautiful orchestration of perception and memory that's happening within these face patches.
Wow, and is this formed in the early stages of development?
Yes.
I know when you're exposed to certain kind of types of faces in your early development, you are able to distinguish them more easily.
Exactly.
But if you're not exposed to certain kind of races, for example, it's more difficult for you to distinguish them later in life.
Yes, yes. The IT cortex, this part of the brain where these face patches are located, it's really wired to learn the statistical structure of what an animal sees, right? And it can change if you see different things. And so these face patches definitely develop over time. They're not genetically determined.
Got it. Very awesome. Has your work been impacted by large language models?
Yes. I mean, the whole revolution in AI has just given us all these new tools to describe what's happening in the brain, right? Because the brain is using these distributed computations where all these neurons are involved, and it's really hard to have intuitions about that. But now we can compare to these neural networks and directly say, oh, this representation maps onto that. And we're especially interested in these video generation models, right? Where you have some text prompt and you can generate a picture from it, like DALL-E.
Yeah.
And we think that these face patches actually implement something similar. So we're very excited about looking into that analogy.
So how does your work interface with BrainMind and AI and brain-computer interfaces?
Yes. So we're trying to understand the neural code for representing the world. But how are we going to test our understanding? How do we know if we got it right? So there, brain-machine interfaces are going to come into play because we want to then stimulate the brain and write in different codes and see what happens. Right? So we're actually developing this technique called holographic optogenetics that lets us use light to activate very specific populations of neurons, and we can actually just add single action potentials to specific neurons. So we can really play with this code and do read-write with very, very high temporal and spatial resolution to test our understanding.
Wow, in what kind of animals are you doing that?
We're doing all our experiments in macaque monkeys.
So doing this optogenetic work as well in...
Yeah.
That's incredible. Wow. Yeah, and what do you hope to get out of BrainMind?
Oh, I've already gotten so much out of it, just, learning such a broad array of ideas, right? From, I think on Saturday, there were talks about death, which I took, personal wisdom from, it's not something I usually think about, all the way to, things that are closer to my interests and brain stimulation, what is the latest. And I should say, it's a chance to meet all these philanthropists you don't usually meet as a university professor. So I really hope that some of them may be inspired by our mission and maybe can contribute in some way.
Yes, have you gotten philanthropic support?
Yeah, so we were very lucky. Fred Ersam is interested in our brain stimulation project. So yeah.
Amazing. So this has funded part of your research. That's incredible.
Yeah, one other thing that I really have loved about BrainMind is that it really forces us to get out of our comfort zone. None of us know or have ever been in this kind of situation. You listen to talk and then you hear about a new artwork. And so it's really been refreshing.
Yes, amazing.